Okay, I think the lecture is being recorded now, so I'll just share my screen with you all. Just a second. So hello everyone, as you all are aware that the present lecture deals with primer designing for cloning CDS region of a gene with either an N-terminal fusion tag, which is a flag tag in our case, or a C-terminal fusion tag, which is a histidine tag in our case, in mammalian expression vector pcDNA 3.1. So before we start the lecture, let us first understand that what is cloning CDS of a gene. So objective of cloning CDS or the coding sequence of the gene is always to express the protein product of the gene in whatever expression system you are dealing with. Right here, since we are stressing upon mammalian expression vector pcDNA 3.1, that means that we want to clone a gene, the series region of a gene, and we want to express the protein product of this gene in a mammalian host. So before we actually start, So I hope the slides are visible and the sound is all right. Can one of you confirm? So I'll just continue playing the slides.
Yes, ma'am. It's visible and we can deliver.
Start designing primers, let's just first understand a few basics. So as we all know that DNA is a double-stranded molecule. It is divided into a plus strand, which is the this strand 5' to 3' strand, and a minus strand, which is 3' to 5' strand. So the thing to understand is that during transcription, what happens is that the minus strand or this strand, which is also known as the antisense strand, it serves as the template strand and codes for the molecule of mRNA. So the molecule of mRNA which is synthesized is perfectly complementary to this strand, which is the minus strand, meaning that mRNA is just a carbon copy of 5' to 3' plus strand of DNA, just the difference being that the T in the DNA will be replaced by U in the mRNA. So because we are dealing with coding the protein of a gene, it is very necessary for us to understand that between these two strands, which serves as the template strand for the synthesis of mRNA, and which strand is in sequence similar to the mRNA, just the difference being that the T in DNA gets replaced by a U in mRNA. So the minus strand or the antisense strand is the template strand, and the plus strand or the sense strand 5' to 3' strand is the strand, the sequence of which is similar to the sequence of mRNA, right? So now when we have to amplify the CDS region of the gene for cloning CDS of a gene, what we do is we use cDNA molecule as the template for the PCR reaction. So let us briefly understand what is a cDNA molecule. We all know that the genomic sequence in the DNA consists of an assembly of exon and intron. When it is transcribed to RNA, then it is just the exons are joined together and the introns they get spliced out. So what we do is for cloning the CDS region of the gene, we extract RNA from a cell, and then we uh, transcribe sorry then we do reverse transcription, in which what happens is that this mRNA molecule gets transcribed into a cDNA molecule, right? So the cDNA, understand this, that the cDNA will only consist of that region of DNA which is cloning for the which is coding for the protein, right? So because introns got removed in RNA, exon got joined together. So the exon joined together would be AUG, which is the start codon, and it will end at UAA, UAG, or UGA, which are the stop codons. So then we isolate RNA from the cell, and then we reverse transcribe it into a cDNA molecule, which will start at ATG and it will end at a TAA, or the stop codon, and we will use this cDNA as the template for our PCR reaction. Now we understood what the template for the PCR reaction would be. Let us try and now understand that the two primers that we'll be using for PCR reaction, where would they bind in our template? So, so as we all know that a PCR reaction will require two sets of primer, right? One is the forward primer and the other one would be the reverse primer. So the forward primer is this one. It binds at the minus strand or the antisense strand of the DNA. So the sequence of forward primer would be similar to the sense strand of the DNA or the plus strand of the DNA because the two strands are complementary to each other. So, right, so as has already been told to you that an ideal primer is 18 to 25 nucleotide in length. So, the forward primer would be the sequence of the plus strand first 18 to 25 nucleotides from the 5 prime to the 3 prime end. Now comes the tricky part, which is the reverse primer. So please always remember that a DNA sequence is always written with the 5 prime to 3 prime polarity. So whenever you are writing down a sequence of DNA, please ensure that you write it down in the correct polarity. Otherwise, the whole experiment will be, the whole experiment will not work. So when I say that the reverse primer binds to the plus strand of the DNA, right? So that means the sequence of the reverse primer would be similar to the minus strand since the two strands are exactly complementary to each other. So the reverse primer would be, since the PCR proceeds in the 5 prime to 3 prime direction in this direction, so the reverse primer would be the sequence of exactly similar sequence of the minus strand. The first again 18 to 25 nucleotides in the 5 prime to 3 prime direction, right? So the reverse primer binds here. That means the sequence will be similar to this end in the 5 prime to 3 prime polarity. So like in this case, when you write down the reverse primer, you will write down as T T A and so on, right? In this direction. This is the 5 prime direction and this is the 3 prime direction. So always remember the polarity of the primer molecule. Now, since the basics are already clear to us, let's start designing the primers. So this all, I think you all are aware that how to retrieve the sequence of a gene. So I will skip all this. So let's come to, so what we now have is the, this region is the, from here to here is the CDS of the gene, right? The coding sequence. So what we'll do is we'll just copy this highlighted sequence and we'll paste it here. So this is a gene, this is a software which is known as Gene Runner. So what we'll do is we'll open Gene Runner. So it is a freely available software. It is compatible with Windows. I'm not sure whether it is compatible with Mac or not. So what we'll do is we'll open Gene Runner and then we'll go to this icon, which is a new DNA sequence icon. We'll open this and you will get a window like this and you will paste your sequence into this window. So when you paste it, you will get something like this. So the benefit of using such softwares is that when I showed you the CDS sequence, the highlighted sequence that I showed you 2 slides ago, so that was what? That was just the plus strand, right? The 5 prime to 3 prime strand. Now when I copied this strand, into software like this, what happened was that it gave me the reverse complement, right? So this is ATG is the plus strand that I copied it, the highlighted sequence that I showed you 2 slides ago, and the strand which is written below is the reverse complement of it. So it will be very nice when we'll make the reverse primer. So what, you don't have to actually, write it with a pen and paper, the plus strand, and then you have to write the reverse complement of it. The reverse complement is already ready with us in the form of this strand. Another use is that we know that I mean the primer, it is not that you just take the 15 to 25, the first 15 to 25 nucleotides from the 5 prime to 3 prime region and your primer is synthesized. So that all the essential characteristics of a primer is already mentioned to you in the previous lecture, so I will not tell you those in details. But what we'll see here is that just taking 15 to 25 nucleotides from the 5 prime end is not primer designing, right? So then, very one important very feature of a primer should be that the sequence should not be self-complementary, right? So let us say if this is your primer and let's say this part is complementary to this part, what will happen is it will bend on itself and form a loop like this, right? So when it has formed a loop like this, what will happen is it will not bind to the template strand. Since the whole part is complementary to the template strand, if it forms a loop like this, one thing is that it has formed a secondary structure, hydrogen bonds are formed, so this will be stable. Other thing is it will not be open to bind to the template region. So all these things, software such as Gene Runner, or you can use SnapGene as well, they tell you, if you put in a DNA sequence in such softwares, they will tell you that whether the sequence is self-complementary, whether it is forming a secondary structure, what is the TM of the sequence, and everything. So using these softwares, it just makes life easy. So as I told you that it is very important to see that the primer is not self complementary. So another thing is that let us suppose that the primer is self complementary, and it is forming a loop like this, but the TM of this loop is, let's say, 15 degrees Celsius, right? So the TM is the melting temperature of the loop that is being formed by the primer. So now you know that PCR is a thermal cyclic reaction, and the temperatures that are there in the PCR reaction are 95 degrees Celsius, which is the denaturating temperature, 72 degrees Celsius, which is the extension, and annealing temperature, which ranges from 50 to 60 degrees Celsius, or maybe 70 degrees Celsius, right? So I told you that my primer is self-complementary and it is forming a loop, but the TM of this loop is, let's say, 15 degrees Celsius. So if the melting temperature of this secondary structure is 15 degrees Celsius, it is it is understandable that this structure will not be stable at temperatures which are as high as 95 degrees Celsius, 72 degrees Celsius, or the annealing temperature, which is generally 50 to 60 degrees Celsius, in the range of 50 to 60 degrees Celsius. So then, if a loop is getting formed in the primer, but the TM or the melting temperature of the loop is less than 15 to 20 degrees Celsius, we can still work with the primer, because at temperatures which are as high as those that are used in the PCR reaction, the loop will be unstable and the primer will open up. So let us now see. What I did was I selected the first 20 nucleotides from the 5 prime to 3 prime end. So this will be my forward primer, right? And then I went to this. Okay, so this is the oligo analysis tool, right? I click on this icon and I get a window like this. Now let us see that what are the features of this primer, right? So the length is all right. The length is 20 nucleotide in length, which is in the range of 18 to 25 nucleotides. TM is also all right, 66.1. So 66.1, if it is the TM, then the annealing will work at, let's say, in the range of 55 to 60 degrees Celsius, or even let's say 60 to 63 degrees Celsius. And the percent GC is slightly high, 65%. So the percent GC is ideally 40 to 60% percent GC content in a primer sequence is all right. But you see the major problem, which is highlighted here with this primer, is that it is forming a hairpin loop. So the oligoanalysis tool of Gene Runner tells you the TM of the primer, the length of the primer, percent GC, and also if it is forming any secondary structure. So we see that it is forming a hairpin. It is forming a hairpin loop. So what is happening is that CCA. So that means this sequence CCA is complementary to TGGC, right? So it is complementary to TGGC, and it is bending, right? Because of this self complementarity. So what happens is whenever the primer is rich in GC, right, we saw that the percent GC is 65%, then the chances of forming a secondary structure is very high, because you know that G and C, they bind with three hydrogen bonds and the binding is very strong. So That is also a hint. If you see that the primer is GC rich, or the sequence against which you need to synthesize the primer is highly GC rich, then there are problems of secondary structures getting formed in the primer. And so one thing is that a secondary structure is getting formed, and the second thing is that the TM of the secondary structure is very high, as high as 45.8 degrees Celsius. So that means that we cannot work with this primer, right? Because 45.8 degrees Celsius means that this primer can still be stable at temperatures ranging from 50 to 60 degrees Celsius, which will be our annealing temperature. So then what I do is I just delete a few nucleotides from this end, right? So now I took just the first 18 nucleotides, and we see that the TM again is all right, 58.9 degrees Celsius. The percent GC is still within the range, 40 to 60 degrees Celsius, and we see no hairpin is getting formed, no dimers are getting formed, no bulge loop. So no secondary structures are getting formed, and the TM is also in the compatible range. So that this means we are done for the forward primer. The first 18 nucleotides in the 5' to 3' strand is our forward primer. Now, two, three things that I would still like to stress upon is that whenever you are using a software like Gene Runner, you should always mention. So the defaults for this software are all right for us. Just that you should always set the probe concentration to 10,000 picomole, right? So the software by default it sets it at 100 or something, but you should always change the defaults and make probe concentration 10,000 before synthesizing the primer, right? So I think we are done for the forward primer. So another thing about primers is that the TM of the forward and the reverse primer should not differ by, let's say, more than 3 degrees or maximum more than 5 degrees. Why? Because we want both the primers to anneal at a single temperature to the template, right? So so now forward primer is synthesized. While synthesizing reverse primer, we should note that the TM should be within so the TM for forward is 58.9, so the TM for reverse should be within 55 to 60 degrees Celsius, right? So that the difference is not much. Now let's look at the reverse primer. So as I told you that the reverse primer binds to the plus strand of the DNA or the sense strand of the DNA. So the reverse primer is 5' to 3' sequence of the antisense strand. So that means TTA, GTT, CCTC, right? So this is how you write the reverse primer, 5' to 3' of this strand. So TTTA, so this is TT TTA, right? GTT, so GTT. So while writing the reverse primer, it is very essential for you to remember the polarity. Please make sure that you write it in the correct polarity, 5' to 3', right? So now let us look at the characteristics of this primer. So the TM of the forward was 58.5. This TM is 57.5. So it is well within the acceptable range. The difference is very slight between the TM of forward and the reverse primer. So they both are capable of annealing at a single temperature. Then the percent GC is also 40.9, which is in the acceptable range. Then secondary structures, we see that two hairpin loops are forming, right? So the software Gene Runner is designed in such a manner that the first hairpin that it shows is the hairpin that is forming at the higher TM. So since first hairpin is forming at a TM of 6.8, the second hairpin would be forming at a TM which is less than 6.8, so we don't have to worry about that. So since I already told you that if even if secondary structures are getting formed in the primer, but if the temperature of the melting temperature of the secondary structure is below 15 to 20 nuclear 15 to 20 degrees Celsius, we don't have to worry about it. So so a hairpin forming at 6.8 degrees, we I mean will not interfere with the PCR reaction or the primer binding to the template, and the length is also 22 nucleotides, which is the acceptable range. So now we have the forward primer with us. and the reverse primer with us. Both of these are in the 5 prime, written in the 5 prime to 3 prime polarity, right? So now we have our primer sequence. The task that remains at hand is to insert this whatever. So the primer sequence, what will it do is, when we'll use the the cDNA of MET30 gene as template with these primers, what it will give us is the CDS region of the MET30 gene amplified, right? The task which now we have to do is we have to take this PCR insert and we need to put it in a expression vector, right? So as the title slide read, that the expression vector that we will be using is the pcDNA 3.1 expression system. So the pcDNA 3.1 expression vector is a vector which is 5.4 base pair in length. So the features of expression vector and cloning vectors, that all you already know, so I will not go into details of this. But what we need to see is that the general considerations that we have to take care while we are designing primers for cloning. So we all know that the multiple cloning site, right, MCS region of a vector is the region where the PCR insert goes and ligates to in a vector, right? So the MCS of a vector, it is its presence is marked by and a vast array of different recognition sites for various common restriction endonucleases. So the MCS of pcDNA 3.1 has recognition sequence for NET1, BMT1, AFL2, HIN3, ACC651, KPN1, BAMH1, ECO-R1, ECO-RB, and so on. So now when we do this kind of cloning, that like the cloning for uh, you know uh, protein expression, what we need to take care is that, as I told you, that the CDS or the coding sequence starts with ATG and ends at TAA. So starts with start codon and ends at stop codon. So what we need to ensure is that it will when it is ligated into a vector and expressed in a host, it is always cloned in the direction where it is starting at ATG and it is ending at TAA. If suppose you cloned it in such a manner that the orientation got reversed, right? So this is ATG TAA, but the clone got inserted in such a manner that it got inserted at as TAA first and ATG last. So what will happen is, during translation, the tRNA assembly will encounter a stop codon at the will encounter the stop codon at the initial step, and protein synthesis will not happen. So what we need to ensure is that this is after the promoter. What comes in the vector is ATG, and what terminates before the poly A signal sequence is the TAA, right? So in order to ensure this, what we do is we perform directional cloning. So directional cloning means that ATG first and TAA last. So how will we ensure that the clone is inserted in this direction only is, we will use restriction endonucleases. We will perform sticky end cloning. So that is also known as sticky end cloning or sticky end digestion mediated cloning. So what happens is, what we do is, we let us take two restriction endonucleases. For example, let us take hint and let us take BAM, right? So what we do is, if we digest this pcDNA 3.1 with hint and BAM, and if suppose in the primer sequence we have hint in the forward primer and BAM in the reverse primer, so, and we digest the PCR insert also with hint and BAM, what will happen is, the vector will also have sticky ends compatible, like the vector will have sticky ends for hint and BAM, and the PCR insert will also have sticky ends for hint and BAM. And then what will happen is, when we perform ligation, the sticky ends for hint will go and bind to the complementary region, or the sticky end for hint. The sticky ends for BAM will bind to the complementary or the sticky ends for BAM. So the sticky ends for BAM in the PCR insert will obviously ligate to the sticky end for BAM in the vector and vice versa. So what will happen is that the clone will be inserted in a particular direction, and you will have a, a recombinant pcDNA 3.1 which can code for the protein which is the product of MET30 gene, right? So you can also refer to this slide, just a reference slide, that vector. So in case of sticky end cloning, that the vector have sticky ends generated like this, and the insert has complementary sticky ends. So when you put them together, they will ligate, and obviously this insert cannot ligate in the opposite direction. It will always ligate like this in this direction, so it will have the start here and it will have the stop here, right? So another advantage of sticky end cloning is that if let's say you digested the vector with restriction enzymes that are cutting in a manner that is generate blunt ends, then the vector has a probability of ligating, forming a self ligated vector, right? So in case of sticky ends, since these two ends cannot join, and since these two ends cannot join, this end can only ligate here and this end can only ligate here, the chances of forming self ligated vector is also less. So then we saw that we need to choose two restriction enzymes that are present in the MCS of the cloning vector. Now the question is that how will these restriction enzymes digest our PCR insert? So the answer is that first of all, we will choose such pair of restriction enzymes, the recognition sites of which are not present anywhere in our insert, right? So the region which we have amplified by PCR previously, for which we designed the primers, we should choose such set of enzymes that are enzyme for such set of enzymes for which the recognition sequence is present in the MCS of the cloning vector, but the recognition sequence is absent in the PCR insert. So how we can do this is that the vector map is already with us. So we saw it previously. What we'll do is we'll paste the CDS sequence. So the same sequence that we pasted in Gene Runner, that is the sequence that we are amplifying. We'll paste the sequence in a software. So this is an online tool. So you just Google NAB Cutter and you will get it. So we'll open NAB Cutter, we'll paste the CDS in this dialog box, and then we'll just click submit, right? So then when you submit, then we'll get, so NEB cutter, what it does is it gives us a list of all the recognition sites that are present in a entered sequence, right? So we'll just go and so this is a bit confusing and it is not clear, so we'll just go on this option that it will give a list of all the enzyme sites that are present, and we'll go to single cutters, right? So when we go to single cutters, we'll get a list of all the enzymes for which the recognition sequence is present in the entered sequence, which is the CDS of MET30 gene, which we have used as the input. So now I've not included the complete list, but what I can tell you is that uh, that in the CDS region of MET30, the enzyme sites for HIN3 and EcoR1 were not present, right? So let us just go again to the vector sequence. So we see that in the MCS, I told you that we can choose any two enzyme sites, right? One in the forward primer and the other in the reverse primer. But the the enzymes should be as such that the recognition sequence is not present in the PCR insert that we are using for cloning, right? So HIN3 and EcoR1 are two enzymes that we're going to work with today. I told you that the enzyme sites for HIN3 and EcoR1 both are not present in the CDS region of MET30. So then so as to insert the PCR sequence, or sorry, so as to insert the PCR product in the correct orientation, what we need to do is we need to put HIN3 site in the forward primer and the EcoR1 site in the reverse primer, right? So how will these enzymes cut? Because in between the PCR product, the recognition sequence for Hint3 and EcoR1 is not present, which is desirable also. But then how will Hint3 and EcoR1 cut the PCR product to get us the desired ends? Is they will do so because we'll put the recognition sequence for Hint3 and EcoR1 in the primer sequence that we are designing. Hint3 in the forward primer and EcoR1 in the reverse primer, right? So then, okay, so I told you Hint3 in the forward primer and EcoR1 in the reverse primer. So the choice of using which two pair of enzymes that we'll use is it also depends upon the compatibility of the two restriction enzymes. So by compatibility, I mean you know that enzymes they work at optimal conditions of temperature, of salt concentration, of buffer concentration. So similarly for restriction enzymes as well, like Hint3 will work in a particular buffer, EcoR1 works in a particular buffer, right? So since we need to perform double digestion, it is ideal that we choose such set of enzymes that are both compatible in the same buffer, right? So that we can get away with digestion in a single step and we don't have to do a multi-step digestion wherein we first digest it with one enzyme, then inactivate the digestion by this enzyme, and then we do the second step, digestion by second enzyme. So for this, what we'll do is we'll go to another online tool, which is double digest calculator from Thermo Scientific, or you can also go to Double Digest Finder, which is from NEB, right? New England Biolabs. So when you open Double Digest Calculator, you will get a window like this. We'll enter the two enzymes, which in our case is HIN3 and EcoR1, right? So when we enter Hint3 and EcoR1, we get a table like this. Okay. So this table will tell us whether these two enzymes are compatible or not. So how can we see this is, if you see these are the different buffers, right? That comes with commercially when we purchase these enzymes, these are the common buffers that comes with them. Okay. So this is a blue buffer, green buffer, orange buffer, red buffer, and Tango, which is a common buffer. So if you're buying enzymes from New England Biolab, you will get buffer 1, buffer 2, buffer 3, which is the common buffers, which in case in this case is the blue, green, orange, and red buffer. And there is one buffer that works with, I mean, it is not mandatory that it will work with all the enzyme, but there is one general buffer that NAB also supplies, that is known as buffer 3, right? And it works with most of the enzymes that are commercially available with NAB. Similarly, the Tango buffer is the buffer that comes with enzymes if we buy the enzymes from Thermo Fisher or Fermentas, and Tango buffer also works with all of these enzymes. So now this is the enzyme activity table. So we see that Hint3, so the incubation temperature for both of these enzymes is 37 degrees Celsius. They both work at 37 degrees Celsius, which is good. We can perform the double digestion in a single step. The recommended buffer for Hint3 is the R buffer. So we see that R buffer give 100% activity for Hint, and for EcoR1 it is the specific buffer EcoR1, which is the unique buffer, right? Or we can also do this in orange buffer. Now units for overnight incubation, it is just if you're performing the digestion overnight, zero point 0.1 units of N3 you should use and zero point 0.2 units of EcoR1 you should use. And thermal inactivation is the temperature at which both of these enzymes will cease to work is 65 degrees Celsius for both. Now we come to this table, which is of particular interest to us, that if we are performing the double digestion, if we say that these two enzymes are compatible, how can we comment that? Is by looking at this table and observing if there is a single buffer in which both of these enzymes work without any star activity. So I hope you all are aware what is star activity. And star activity, uh, so star activity is just briefly that if you're not using optimal conditions for performing a restriction digestion, it may so happen that the restriction enzyme will cut at sites which are not its recognition sequence. So it can perform non-specific cuts. So wherever you see, so you see a small star which is here. So this star denotes that in R buffer, which is the red buffer, eco R1 shows a star activity, right? So eco R1 will perform unspecific cuts if you incubate the digestion with EcoR1 in a red buffer system. So, like, blue buffer does not work for both of these. Green buffer does not work with EcoR1. Orange buffer does not work with Hint3. R buffer may EcoR1 is having star activity. Tango buffer, if you're using it at 1x concentration, EcoR1 will not have any activity. While if you use Tango buffer at 2x concentration, we see that EcoR1 has 100% activity and Hint3 has 50 to 100% activity. So that means that these two buffers are compatible, as in the digestion can be put in Tango buffer at 2x concentration so as to achieve a successful digestion. What one minute thing we'll have to do is whatever amount of EcoR1 we are putting, since EcoR1 is having 100% activity, we'll put slightly more amount of Hint3 or 2 times Hint3 because it is having activity in the range of 50 to 100% in 2x Tango buffer, right? So now our primers were ready. Now we know that what we can take the decision of what restriction enzymes we can choose, and we saw that whatever restriction enzymes we thought that we could work with, they're also compatible. Double digestion can happen in a single go in a single buffer with these two enzymes. So we'll design the primers with the Hint3 and the EcoR1 site. So the Hint3 site is AAGCTT, which is in the forward primer because the vector was oriented in such a manner that if you don't put Hint3 in the forward primer, you won't have the clone in the correct orientation. And the EcoR1 site, that is GAATTC, is in the reverse primer. Now you see this that I have highlighted. These three nucleotides are known as overhangs for efficient digestion. So what happens is that you know that all these enzymes, not just restriction endonucleases, but whatever enzymes you're working with, they all have a tertiary structure and they are active only when their tertiary structure is restored, right? So the restriction enzymes also they have their particular tertiary structure in which they are active, and in order for efficient restriction to for efficient digestion to happen, the enzyme should sit at the DNA sequence properly, right? The problem with this kind of restriction digestion is that digestion site is added at the terminal of the sequence, right? So that means if your sequence is like this, digestion should happen at this place and at this place. So that means since these are the end of the DNA molecule, there will be no place for the restriction enzymes to come and sit. So it may so happen that the binding of the restriction enzyme to the sequence is not efficient and they fall off. If they fall off, the digestion will obviously will not be efficient, right? So in order to prevent this, what we do is we add nucleotide overhangs right at the end of the primers. So how many bases you should add is also a decision that you have to take. So it depends upon the restriction enzyme that you're using. The commonly used restriction enzymes like N3 EcoR1, BAMH1 EcoR5, KPN1, MLU1, all these enzymes, they work best if you add a 3-base overhang. But the choice of the number of bases that you should add as overhangs depends upon the enzyme that you're choosing, and this you can check on again NEB website, New England Biolab website. So if you just Google overhangs for Hint3 NEB, you'll get the NEB website wherein they have a table where they show that how many nucleotide overhangs should be added for each enzyme. So in our case, for Hint3 and EcoR1, a 3-base overhang is sufficient. So I added TAT and ATA. So these three nucleotides can be anything, XXX, but we generally choose not to add a G or C here because again, if we add a G or C here, it may so happen that the whole primer becomes self-complementary, the GC content rises. So we try and not add GC, we try and add a random sequence of A and T. So TAT in the forward primer, ATA in the reverse primer, or you could have done ATA in the forward primer, and I mean whatever. It's just a random sequence of nucleotide, 3 nucleotides, and preferably A and T. So now we have the primer sequence with the restriction site and the overhang. So if you were to clone just the CDS of the gene, the primer sequence that you saw in the previous slide was sufficient, and we need not proceed any further. We'll just order these primers and the work is done. But lecture also uh, the lecture also covers cloning of the CDS along with a fusion tag. So fusion tag means that whatever, like I told you that this cloning particularly, it will lead to the expression of protein product of MET30 gene, right? So let's say if you want MET30 protein to be tagged to a fusion tag or tagged to something, so the application for adding a fusion tag can be many. One very simple example I'll give you. Let's say if you want to study the localization of MET30, if you want to see in which whether the MET30 is present in the nucleus, whether MET30 is expressed in the cytoplasm, whether MET30 is expressed in the mitochondria, or just the localization of MET30 protein. What you can do is you can add a fluorescence tag to the protein, right? So you can add like GFP is a green fluorescent protein. So what you can do is you can fuse the sequence for MET30 CDS along with the sequence for GFP CDS so that they are in frame, so that they are translated together. And then what we'll have is you will have a GFP tagged with the MET30 protein. So wherever MET30 is getting expressed, along with it, GFP is also getting expressed. They're getting expressed as a single protein, right? So now if you want to study MET30's localization, it is very simple. You can just visualize the GFP and you will know where MET30 is getting expressed in the cytoplasm, in the nucleus, or in the mitochondria. So the purpose of adding fusion tags can be many, but I just gave you a simple example for your understanding. So in this lecture, we'll add a flag tag, right? So this is the sequence of flag tags. So this particular slide, I'll tell you how to add. So if you want to add a N-terminal tag, right? N-terminal fusion tag, that is the flag tag. So N-terminal tag means that the tag will express first and then your protein. So the protein will be expressed as one, but first will be flag and then your protein will start. So adding N-terminal fusion tag is very simple. What we'll do is we'll just put this flag sequence in the forward primer. And if you see that I told you that the protein, this flag will be fused to your protein, right? So we ATG is already present in the tag, right? That is the forward start codon. So we'll just omit this ATG in the forward primer, which I've done over here, right? Because protein will start from here. First flag will be expressed and then your protein. So having N-terminal fusion tag is pretty simple. Adding the sequence, we'll just add it in the forward primer. Now, what
And
this again, I like to draw your attention to. This is a Kozak sequence. So Kozak sequence, we all know that it is like Shine-Dalgarno sequence is present in case of prokaryotes. Kozak sequence is present in case of eukaryotes. So it is present upstream of the so when you're adding it in the so there are certain vectors that already have the Kozak sequence in them, then you need not put this Kozak sequence in your primers. But there are certain expression vectors which does not carry the Kozak sequence, and you put Kozak sequence upstream of your primers to ensure efficient translation, right? Then so this is the N-terminal fusion tag. I've just added this sequence. Now one important thing that we need to take care is that we need to check whether the like I told you that so protein synthesis, it happens from start codon to stop codon. So ATG, TAG, TAA, right? So this is three nucleotides code for a single amino acid. So all this has to be in frame, right? Let us say that ATG is coding for methionine, and then you omit one A from here. So methionine GAT, let us say if it was coding for an amino acid X, now GAT, since A is deleted, it will code for another amino acid which is GTT, right? So framing of the it is very necessary that whatever tag we are adding, it is getting expressed in frame with our protein of interest. So how we can this is, we can use this tool, which is Snapgene Viewer. So if you open Snapgene Viewer, if you download Snapgene Viewer and you open it, you will go to new DNA file. So you'll get a window like this. Here you will enter your sequence. So right, what I'll enter is I'll enter the my primer so I'll enter my primer sequence along with rest of the CDS, and I'll just click OK. Then this is the sequence. So this is my primer sequence. So primer sequence, I will just remove the restriction enzyme sites, because obviously that is not coding for a protein. Protein will start from ATG, so I will include the primer from ATG, and the reverse primer as well. I will omit the overhang, I will omit the restriction enzyme site, and I'll just use the reverse primer that is terminating at the stop codon, right? So then I will go to this option, which is translate, and I will translate it in the so when you go to this option, when you go to this icon, you will get a series of options to translate, and you will translate the top three frames. So top three frame means first frame will start from this ATG, second frame will start from TGT, and third frame will start from GTC, right? So top three frames we will translate, and we'll see if it is getting translated properly. So when I do this, so this is the top three frames. So first one starting from ATG, we see that it is coding for methionine, which is this what is the purpose of start codon. Then TGG starting from here, and then GGA starting from here, right? So we are not concerned with these two. We will only check this because this is what we want to clone. So we see that this is, so the Snapgene has a very interactive feature. It will, if it recognizes certain special features in your sequence, it will tell you. So it recognizes that it is coding. This is a flag tag that your sequence is coding for your primer sequence. So it has added a flag here, and then in frame with, so you see in frame with flag is your protein getting expressed, and then we see that it is getting expressed. So the star donates a stop sign, stop codon, right? That the translation will stop at this point. But you see that there is no star in our sequence in the topmost frame. So it is showing a star only at the end, which is the TAA, or the stop codon, right? So that means that it is in frame also. All right. So that means our primers are ready, right? So that means our primers are ready. Whatever sequence I showed you is the sequence that you will use if you want to clone MET30 with an N-terminal flag tag. Now we come to cloning the same protein with a C-terminal tag. So sometimes what happens is, let's say a certain protein is destined to be expressed in nucleus, right? So what happens is, proteins are synthesized in the cytoplasm, they get processed in the endoplasmic reticulum, and then they travel to whatever their destination is. So certain proteins that are destined, let's say, to be present in let's say nucleus, so they carry an N-terminal nuclear localization signal, right? So in case of such proteins which have the signal sequence at their N-terminal, adding a flag tag or any other tag may disrupt the signal sequence. So in that case, the need, if you still want your protein to be fused with some tag, then you will do so at the C-terminal region, right? So for having a tag at the C-terminal, I think it is now pretty clear that the uh, tag sequence will be added at the reverse primer, right? Now, as I told you, reverse primers are always tricky, so adding a C-terminal tag is also a bit tricky. So in our case, in this case, we are we're dealing with histidine tag. So I'll just tell you the normal histidine tag sequence. So in 5' to 3' direction, histidine tag is CAT. So we add six histidine, six S, okay, six histidine. So CAT is the codon that codes for histidine. But since you have to add it at the C terminal, you will need to add it in the reverse primer. Then how will you add it? You will add it as, so CAT means The reverse complement of this is GTA. So then your reverse primer, so what you will do is you will just, so your stop codon was here initially, you will remove stop codon from here, you will put the stop codon at the end, and in between the rest of the sequence and the stop codon you will place your tag sequence, and the reverse complement of tag sequence you will place. Please remember, so reverse complement will be GTA, right? So this is 5 prime, this is 3 prime. So when you synthesize, when you go like this, GTA, so you will write it in the 5 prime to 3 prime direction will be GTA, 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 and 6 times the same sequence, right? So now this will be your primer sequence if you want to put a C-terminal histidine tag. So please see that your forward primer is now reduced to just the restriction enzyme site, overhang, and the 15 to, the first few nucleotides of the sense strand. Whereas the reverse primer is now the overhang, the restriction site, stop codon, fusion sequence, and the primer sequence. So that's it. Uh, so the cloning of MED30 CDS with N-terminal flag tag, you have this sequence. Please remember that Kozak sequence is not a mandatory feature of such primer. If the vector does not have a Kozak sequence, then only we'll add. Otherwise, we don't need to. And the primer set for cloning a CDS with the C-terminal histidine tag is this. So I hope that things are clear in your head. But still, if you have any doubt, you can always write back to us.
So yeah, that was the lecture. I think ma'am has shared the slides with you all also. So are there any doubts?
Excuse me, ma'am. I had asked Ritu ma'am to share instructions on what we are supposed to submit and what we are supposed to work on for the assignment.
I
So,
can't, I can't,
can you hear me, ma'am?
uh-huh.
So, ma'am, I had asked Ritu ma'am in a previous lecture about what we are supposed to be doing in these assignments and what we are supposed to submit. So she had said that she'd send an email, but we had not received any email yet.
That ma'am will send you the email in due time. We'll just remind her. And I think that question ma'am can answer better
Okay, ma'am. Can you please just ask her so that she can tell us tomorrow?
to her.
Okay, thank you, ma'am.
Hey guys, got a few emails, but sorry I could not reply earlier. But I'll upload the PPT in Teams only. Okay. Okay.
So any more questions?
Hello.
Yeah.
So, ma'am, can you show how to download the CDS sequence?
That was shown in the last lecture, no?
Actually, ma'am, I have tried to download the CDS sequence, but it is not matching with what you shown right today.
So
Actually,
there are three transcript variant, right? Did you download the longest?
ma'am, I have downloaded the three. Uh,
Okay, then let me show you.
कोई भी कर लो.
So I hope my screen is visible to you all. Is my screen visible to you all?
Yes, ma'am.
yes, ma'am.
So you go to this database gene, enter whatever gene, whatever is your gene of interest, the name of the gene. Then you go to the desired uh, organism. Like in our case, we work with Homo sapiens MET30. So I will just open this one. Then you scroll down, you will find a list of options. So mRNA and proteins, and you will see that you can see that there are three transcript variants. Out of the three, I chose this one, okay, because it is coding for the longest isoform.
Then isoform third you have chosen last time.
Yeah.
Now you are isoform one or third.
Isoform third, I told you that whatever is the longest isoform, we work with that. If we don't have a prior knowledge of which isoform is getting expressed in particularly which type of cells and what is our objective of the study. So if we are not sure whether we want to work with a particular isoform, we usually go with like in case of no prior information, we will go with the longest isoform.
Okay.
So you go to the CDS and there you have the sequence.
1 to 961.
Okay, there you have the sequence.
And ma'am, how to sir means color the sequence like brown? You have colored.
I have not colored. I just clicked on the CDS, so it has the tool has highlighted the CDS region. Okay,
Okay.
there is no coloring. You just select the CDS, so it will highlight the region that is coding. That is the CDS sequence of the gene. So this is just a highlight that is made by the software itself.
But in my tab, it is not happening like that.
That I'm not so sure why is it not happening. But this is
Okay.
nothing
I'll
special.
try.
If you go to FASTA, na, like if you're not able to select this thing, you select CDS and then go to FASTA.
Okay.
Uh, this sequence is just the CDS sequence, or you can also use a laptop or a computer to do it.
Okay.
So any other question?
And
Yeah.
in the last section where we had added the tags to our sequence of interest, so in case when we shifted the stop codon,
Hmm.
so for that was for translational fusion. But if somebody wants to do a transcriptional fusion, then we would put the stop codon in the middle of the CDS, in between the CDS of the tag and the coding sequence of our gene.
No, I'm sorry, I don't understand the question. If you have
I'm
to, huh.
uh, actually, I wanted to understand what the position of the confirm rather, what the position of the stop codon would be if we are trying to do a transcriptional fusion rather than a translational one.
So let me first make myself clear is that if you want, even if you want to express, or if you want to, the objective of your study is a translational fusion. If you are doing so via molecular cloning, you will incorporate the tag sequence in the transcript or the mRNA sequence of the gene itself, right? Because
Yes.
you're cloning means you're amplifying from the parent strand, and parent strand is the cDNA, right? cDNA, we are using it as the template. So if the final
Yes.
objective is that you have to, let's say your protein is there and you want to incorporate a C-terminal fusion tag. So the protein will, so if your protein is protein A and the tag is, let's say tag G, you want protein A and tag G to be expressed together. So what you will do is you will put the sequence of protein A, then you will incorporate the CDS, the CDS, the coding sequence of the tag G, and then you will put a stop codon so that the protein does not terminate after it has or the translational machinery does not terminate after it has cloned for protein A, it will also incorporate the sequence of the tag, which is the protein G or the peptide G, and then stop codon will come.
Okay.
Ritangsha, what do you mean by transcriptional fusion?
Yes, sir. That actually what I meant was that translational fusion. So there, our suppose if we are not using a flag tab, suppose we are using a GFP. So if it is translationally fused with a protein, then wherever our protein is moving in the cell, you, in the same location, our tag is going to move because they are attached together.
Right.
But in case of transcriptional fusion, which we use for certain studies, the tag and the
I
protein.
think I got your question.
Yes,
Uh,
sir.
and
So
if it is a non-coding, see, if your gene is a non-coding gene, then only your question is relevant. Okay.
Yeah.
Like, for example, long non-coding RNA or microRNA, you want to localize where in the cell this particular RNA is, right? Because if it is a coding gene, it will ultimately form a protein. And then it doesn't matter whether you put a transcriptional probe or a fusion protein,
Translational.
translational fusion protein. So if you're asking how to locate a non-coding RNA in the cell, there are many ways to do that. For one example, I would tell you like RNA fish is there by which you can identify, and you need to make some probes for that. So that is a whole different thing. So that is right now out of the scope of our discussion, but that is a different aspect. Yeah.
okay, sir. Thank you.
Pallavi, I think he has a question.
Yes, I have two questions. So initially we started off by making primers which were 18 to 25 nucleotides long, and then we have added a lot of like a lot of bases on at least one of the two primers, which includes the restriction site and also either of the two tags. So do the primers now become longer than 25 nucleotides?
No, yes, obviously they will
So
be.
but then since the like the usual guidelines are made for a primer which is less than 25 nucleotides long, so now if it is like 45 or 50 bases long.
What the objective of uh, this particular primer design was to incorporate a tag. That is why the
Right,
primer sequence was like it was more than 25. It was almost 50 to 60 base pair in length.
right, right,
So the guidelines say that the ideal length of a primer should be 15 to 25 nucleotide in length. That means, see, if you understand the chemistry of DNA, what happens is, or the chemistry of a PCR reaction, that the primer will go and bind to the template, right?
right,
So whenever DNA forms a double-stranded structure, what happens is that structure, now DNA is no more arranged as a primary sequence, right? It has formed a secondary structure.
right,
This secondary structure will have a particular TM, which is called the melting TM of
right,
double-stranded DNA.
right,
Now, the guidelines say that the primer should be 20, 15 to 25 nucleotide in length because, see, TM, if you guys know, remember your basic chemistry lectures, TM is the temperature at which any double-stranded DNA is open. 50% of that strand is in open conformation, right?
yes,
So, if you design a primer sequence which is, let's say, 50 base pair in length, which in our case it was 50 to 60 base pair in length. But that 50 to 60 base pair of the primer sequence is completely complementary to the parent DNA. So what will happen is that the secondary structure that will form when the primer is binding to the parent DNA will have a very high TM. So what will happen is that the secondary structure that is getting formed will be very stable for subsequent PCR reactions, right? PCR is what it is, a cyclic reaction. So one cycle will take place where one DNA will be like 2n hota hai na formula. So what will happen is the DNA doubles in one cycle. Then another cycle will happen. So what will happen is that the TM of the double stranded DNA which is getting formed will be so high that in subsequent cycles annealing will be very difficult. That is why we say that we don't want a primer which is more than 25 nucleotide in length. But here what we have done is that complementary sequence which is complementary to the template is still 15 to 25 nucleotide long only. What we have added is that part of the primer which is not actually annealing to the template, right? Because the CDN, the CDS, the CDNA which we are using as template will not have the sequence of the flag tag, will not have the restriction sites. So in this case, it is like it won't affect the reaction, the chemistry of the reaction that much. But if we are synthesizing a primer which is all 50 base pair or all 70 base pair is complementary to the template DNA, then it creates some problems.
All right, okay, I've understood that. So, but just one more query is that now that since the number of nucleotides have increased, the chances of dimers or self dimers or cross dimers would also increase. So maybe we'll have to do another check on that.
Yeah, you can do, but see, whatever sequence we are putting externally, we take care that they are not so GC rich, they are not so self-complementary. Like flag tag, whatever sequence we are putting,
Mm-hmm.
flag tag is a very well-used, very commercially viable tag, which is put in fusion whenever we are synthesizing fusion proteins. So that
Okay.
sequence is checked.
Since
And another
okay, it's
thing is,
optimized.
yeah, it's optimized. And another thing
Okay.
is that see, by means of cloning, we can only add a small tag to the protein sequence. Like flag tag is a small peptide.
Right.
Even histidine is a small peptide, like 6 histidines we are adding.
Right.
If you want to actually fuse two proteins together, like two huge sequences together, then it is not possible by this method. It is not possible by traditional
Right.
method.
Right. Right. Okay. Okay. Thank you.
So, are there any other questions? So I think the lecture was pretty lengthy and it must have been tiring also. So you can go through the lecture again and you can try designing primer for any specific gene of your interest. And then if you're facing any problem, you can get back to us. You can write an email to us, or in the next class we can discuss before beginning the next lecture. So if there is nothing else, I mean I'll just stop the meeting. So can
Thank you.
anyone? Yeah, okay. So I'll just end this meeting and I'll remind ma'am about the assignment, right? Some assignment ma'am has to send if I understood it correctly. Can any one of you confirm what was the initial query that ma'am had to send some assignment by today, right?
Ma'am, he was talking about an email he did not receive.
So he in particular did not
So
receive the
I
email.
think ma'am has to send us a list of genes that each of us are supposed to work with along the lab sessions. Each of us will get one gene, one
Okay,
different
so
gene.
that list has to be sent by today.
Yes, yes.
Okay, okay, okay. I'll just remind ma'am. Okay, then I'll just stop the meeting. Thank you so much for your attention, and we'll see you in the next class.
Thank you, ma'am.